Hello and welcome to this video where we're going to go through the answers to the questions on pages 61, 62 and 63 of the Separate Science Booklet. Let's get started. So, question 1. Charles Darwin investigated tropisms in plants. Some students did an investigation similar to Darwin's investigation. They grew seeds until short shoots had grown. They then used black plastic to cover parts of the shoots. So here you can see a jacket around the bottom half, and here you can see that the tip is covered, and this one here has no plastic covering it. They then put the shoots in light coming from one direction, which was coming from the right in all of the examples, and they put boxes over the shoots so that there was no other light. The diagram shows how the investigation was set up, and underneath we've got the diagrams where 2 days later they took off the black plastic covers and looked at the shoots, and the diagram shows the results. It's pretty clear that in A and B we can see that they've bent towards the light, whereas in C nothing has happened. So, give two variables that the student should control in this investigation. Well, when we're thinking about this investigation, we're thinking about what things they want to keep the same. So the things that they'd want to keep the same in this investigation would be, for example, the species of plants that they were using. As different species may react in different ways. The other things we'd want to keep that constant would be the age of the shoots. It wouldn't be fair to use one shoot that's say a week old and one shoot that's 3 weeks old, because that would make a difference in those results. Other things that you could think about was the light, so the intensity of light that was coming in, or the colour of light that was coming in. And we also want to think about other variables, so it could be the temperature that the boxes were kept in, the amount of water available to plants, etc. So there's plenty of answers to get yourselves those 2 marks. B. Shoot A bent towards the light as it grew. Explain how. So using our work on hormones, we know that the hormones such as auxin develop on the shady side. So the auxin would be developing on this side here. So auxin builds up on the shady side. Now the build-up of auxin in the shoots causes the cells on that side to elongate. And what that does is that these cells on this side get longer and these cells don't, and that longer side starts to make the shoot bend towards the light. So, what conclusions can be drawn from the results about the detection of the light stimulus? Well, let's have a look at these results. We can see clearly that A and B have changed in response to the stimulus of light, but C hasn't. And the only difference between C and A and B is that both A and B have the tip uncovered, whereas C has the tip covered. So therefore we can draw the conclusion that covering the tip means that they don't respond to the stimulus of light. So the detection of the light stimulus must take place in the tip. Okay, let's have a look at the next question. Where in the shoot the response to the light takes place? Well, we can see that the response doesn't actually take place in the tip, it happens just behind the tip here. So I'd say that it takes place in the top half. If we look at the bottom half of all of these shoots, they all look the same. It's the top half that's different. So where the shoot response to the light takes place, in the top half of the shoot. Okay, let's move on to our next question. Hormones called auxins control plant growth. A student investigated plant growth responses in roots, and this is the method they used. So they grew 3 bean seeds until their roots were 1 centimetre long. They then attached them to moist cotton wool in a petri dish. Each bean seed root should point in a different direction. So this one's pointing down, across and up. They fixed the petri dish vertically for 2 days in the dark. And the figure shows the results over here. Describe the direction of growth of the bean roots after 2 days and give a reason for this growth response. So this first mark's really simple. We just got to describe the direction, and we can see the direction in front of us. This one's gone down, this one's gone across and therefore down, and this one's going back down. So the direction of the root growth is downwards. Now the reason for that is because of what we call geotropism. And that is that the roots are responding to gravity. So they're growing in the direction of gravity, which is why they're all moving down. We know that it's not a phototropism response because it tells us in the question that they're kept in the dark. The student then noticed the shoots growing from the seeds. He then put a light above the petri dish but didn't move the seeds. He allowed the seeds to grow for 2 more days. Predict the direction of growth of the bean shoots after 2 days and give a reason for your prediction. Now again, it's a prediction, but you need to have a sensible reason. My prediction would be that the direction of the root growth would be upwards, and the reason would be that they're growing towards the light, which is what we call phototropism. Ethene is a plant hormone. Ethene causes fruits to ripen. Scientists measure the concentration of ethene found in fruit at different stages of ripeness, and the figure shows their results. At which stage of the ripeness is there most ethene? So again, simply we need to look at our graph. Our axis type on our y-axis is the concentration of ethene. So the higher up the graph you go, the more ethene there is. And we can clearly see that this point here is the highest point on the graph. And if we look down, that is the 3rd stage of ripeness. Suggest how the scientists can find out if the result for stage 1 was an anomaly. Again, these questions are very repetitive and we want to get in the habit of the answers. So to find out if it was an anomaly, you would repeat the results. And if you kept getting continuously answered the same, then you would know that it wasn't an anomaly. Gibberellins are a different type of plant hormone. Farmers growing cotton plants in cold climates sometimes soak their seeds in a solution of gibberellins before planting the seeds. Suggest an advantage of soaking seeds in a gibberellin solution in cold climates. Well, we know that gibberellin promotes the development of plants, and if they're in a cold climate you want to have a longer growing season because they're going to grow slower, and so therefore to get to the same size they need a longer season. So by soaking them you would increase the potential growing season. And so that concludes the questions on plant hormones as part of our Separate Science booklet. Happy sciencing!